Okay, we'll go ahead and pick back up. Now we're still in Chapter 5. This is just the second part of the 5A portion, and this will be placed on your media. But basically what we were talking about was cellular respiration. Cellular respiration, again, is what we see more in a biological system. When we talk about especially microorganisms, we'll see this more commonly in eukaryotic cells. But when we talk about cellular respiration, we're talking about the oxidation of molecules that liberate electrons for the electron transport chain. ATP is basically generated by oxidative phosphorylation. And we talked about some of the intermediate steps that were used in this process, intermediate steps from glycolysis, where pyruvic acid is generated, the breakdown of a 6-carbon sugar, glucose, for example, into 2 molecules of 3-carbon sugar, otherwise known as pyruvic acid, oxidized and decarboxylated. Carbon dioxide is being generated in this sequence of enzymatic reactions, which will lead to the Krebs cycle. Again, references from your textbook, Figure 5.1. But when we talk about glycolysis forms pyruvic acid from carbohydrates such as glucose. Pyruvic acid loses molecules of carbon dioxide, decarboxylation reaction to form an acetyl group. These then will form a complex. The complex is known as acetyl-CoA, where you'll see the acetyl group, the functional group, will combine with, again, coenzyme A, and will enter into the Krebs cycle. The Krebs cycle is, again, a series of enzymatic steps that will continue to break down this carbon compound to produce energy in the form of ATP. So in the Krebs cycle, we see releases of energy, acetyl-CoA, in a series of steps. The first step requires energy. Remember, to get energy, to produce energy, it also requires energy. And we see in this first process, it'll yield citric acid. Other name of the cycle, sometimes you'll see it referred to as the citric acid cycle. Again, I'm not going to ask you to go through each of the steps of the Krebs cycle or to identify the enzymes that are required for each step of the Krebs cycle, but I do want you to appreciate the process. I do want you to know where this cycle takes place at and its importance in that it will produce these high energy compounds, ATP. So subsequent energy yielding steps result in a loss of carbon dioxide and a loss of hydrogen atoms. 4 molecules of CO2 are basically released in the atmosphere, and for every 2 molecules, sorry for the typo, for every 2 molecules of acetyl-CoA, then it enters into the cycle. Most of the energy contained in the 6 carbon molecules of NADH and the 2 molecules of FADH. So in the Krebs cycle, we see oxidation of acetyl-CoA. It produces NADH and FADH2. This is a series of steps that are used in the illustration that's shown in your book. And again, I'm not spending a lot of detail on it because I'm not going to ask you to go through the steps of that process. Again, knowing the Krebs cycle takes place inside the mitochondria and yields energy in the form of ATP. And in the electron transport chain, you'll see carrier molecules that will be oxidized and reduced as electrons and passed down in a series or a sequence of reactions referred to as being passed down the chain. Energy release can be used to produce ATP in the format of chemiosmosis, as we referred to earlier. Now, again, in the electron transport chain, we see energy from NADH and FADH2 generated in the electron transport chain. Again, I'm not going to ask you these steps particularly. Just want you to know that it's a sequence of events, again, where energy production will take place. The energy reduced are used to drive these types of generation, the chemiosmotic generation of ATP. We'll see later on in the lecture. Basically, in eukaryotic cells, the chain is in the mitochondria, and prokaryotic cells will see it in the plasma membrane. Now, some classes of carrier molecules that will carry these electrons are basically 3 classes: flavoproteins, cytochromes, ubiquinones, or coenzyme Q. These are non-protein carriers, as I've alluded to earlier in the lecture. These carriers can accept protons, hydrogen protons, and also and/or electrons. It places the electrons are basically pumped from one side of a cell membrane to another. This causes a chemical charge to build up on one side of the electron. It creates a membrane potential through which we can see this energy being generated and transferred. The buildup of these charges, positive charges on one side, negative charges on the other side, the buildup provides stored energy like you see the example used in your book is water pushing on a dam, which basically will provide energy for generation of ATP by chemiosmosis. Your book has a very good illustration that typifies chemiosmotic mechanism for ATP generation. Again, electrons from NADH pass down basically a chain, again in reaction. Some of the protons are pumped across the particular membrane, and the carrier molecules basically that do this are referred to as proton pumps. Remember, these substances sometimes aren't freely permeable and require energy. A proton pump would require energy to be able to pass these charged particles across. As protons accumulate basically on one side of the membrane, a resulting protein or motive force from the positive charges basically begin to accumulate on one side of the membrane versus the other, and these protons will diffuse back and across particular membrane in specialized channels. This is referred to or is used to synthesize ATP. And again, you can see enzymes are used in that process. Again, the enzyme ATP synthase is used for this potential to be caused across membranes. As you can see in this illustration, it typifies this process by which you can see positive charges will line up on one side of the membrane or another. Again, those of you in AMP 1 and 2 familiar with the phospholipid bilayer, the lipid layers not freely permeable. Typically, it's the protein channel that will allow these charged particles to pass back and forth through the cell membrane. They're not freely permeable through the cell membrane itself. It will have to pass through these protein channels. Again, to the left, you see the illustration of respiration in a cellular or a single-celled organism where glycolysis takes place. Again, glycolysis taking place inside the cytosol. Glycolysis causes glucose to be broken into pyruvic acid, which is a 3-carbon sugar, then into acetyl-CoA, then inside the mitochondria. You see the Krebs cycle take place, CO2 generated, ATP, the energy constituent is produced, NADH is produced. It's a byproduct, and these charged electrons, this energy component is then passed through a series of electron receptors, which can either be organic or inorganic, in the production of ATP. Oxygen is given off, water is given off. These are the simple steps of how carbon substances are used and broke down. We haven't talked about fermentation yet, but there are alternative processes as well where we can see energy production take place. Again, you can see the importance of NADH as an energy component. You see similarities on both sides. You also see a little bit of ATP is produced in the cytoplasm, in the cytosol, by glycolysis. That's important because there is a requirement of energy to be produced. We have to have glycolysis, even though glycolysis takes place in the absence of oxygen. So we don't see cellular respiration as I've talked to before. So glycolysis takes place basically in the absence of oxygen, but it's a part of anaerobic, or in the absence of oxygen, anaerobic metabolism. But equally as important, because we can't get to pyruvic acid and we can't get to the Krebs cycle without the production of these 3-carbon sugars. So anaerobic metabolism is important in our system as well, just as it is in fermentation. But we'll go into more detail about fermentation in just a minute. Chemiosmosis basically again is referring to the electron charges that we see on each side of the cell membrane. Again, the illustration here is referring to the phospholipid bilayer that we see in cells. Again, the phosphorus ends, the lipid ends pointed toward the inside, not freely permeable substances, charged particles that pass through the cell membrane, basically are passing through these protein structures from one side to another. It's the charges, it's the transmembrane potential that develops on each side of the membrane that allows the generation of energy. And you can see as these charged particles are passed through the protein, generation of ATP is the result. So two different respirations. Aerobic respiration typically means presence of oxygen. The final electron acceptor in the electron transport chain is oxygen, aerobic respiration. And anaerobic respiration, the final electron acceptor in the chain, is not O2, it's not oxygen, yields less energy than aerobic respiration does because it's only part of the Krebs cycle can be operated on this anaerobic condition. So when we talk about aerobic respiration, prokaryotic cells, the more primitive cells, generate about 38 molecules of ATP aerobically for each molecule of glucose. That's significant. Eukaryotic cells produce 36 units of ATP. Some bacteria that use oxygen substitutes, such as nitrate oxygen, and certain organisms don't require oxygen. They can exist in different environments. They'll form these nitrate ions, or nitrous oxide or nitrogen gas, sulfates, they form hydrogen sulfides, or carbonates, which forming methane. And so also in aerobic respiration, you can see different electron acceptors can be used. Nitrogen, sulfur, carbon can also be used as electron acceptors instead of oxygen to generate different products. The pathways that we've talked about, glycolysis, you need to know where glycolysis takes place at. In eukaryotes and in prokaryotes, glycolysis takes place in the cytoplasm, inside the cell. The intermediate step, again, a part of glycolysis, we see that takes place in the cytoplasm. The Krebs cycle in eukaryotic cells, or eukaryotic organisms, I should say, and inside the biological system, the human system, takes place inside the mitochondria. In the primitive cells, prokaryotes, we see Krebs cycle takes place in the cytoplasm, and the electron transport chain, mitochondrial inner membrane, and then in prokaryotes, we see that takes place in the plasma membrane. Now, all of that I've talked about before leads to this one illustration. You should be familiar with this illustration. You should know where glycolysis takes place at. You should know the basic step of glycolysis, breaking down glucose into 3 carbon products. You should know the basic steps of the Krebs cycle, which basically take place inside the mitochondria, there to produce energy packets, NADH, and eventually ATP. And then the electron transport chain, where that's located at, why it's an electron acceptor, and again taking place in the eukaryotic cells, in the mitochondria inner membrane, and then in prokaryotes in the plasma membrane. Energy produced, I'm not going to ask you where and how much ATP is produced at any given step. The whole sequence of reactions is for the production of energy. That's what I want you to appreciate. Obviously, you should appreciate, and maybe not obviously, I'll say you should appreciate that anaerobic metabolism does not generate as much ATP as aerobic metabolism, so you should appreciate that aspect of ATP production. Now, fermentation. Fermentation is a little bit of a different process, but it's an alternative pathway that's used, again, to be able to produce energy in some organisms. After glucose is broken down into pyruvic acid, the pyruvic acid can undergo fermentation. So fermentation doesn't require oxygen, or it does, and neither does it require the electron transport chain, as we talked about, that takes place inside the membrane. In fermentation, pyruvic acid accepts electrons, hydrogen, and then it turns into various end products such as lactic acid and ethanol. Now, in these homolactic fermentation processes, what basically is produced is lactic acid. Lactic acid, as a byproduct in the human system, can become toxic at some point, but we'll go into a little more details about that later on. Heterolactic fermentation produces acids and alcohols as well as lactic acid. So you should know the differences in those two types of fermentation, one of which homeolactic producing lactic acid only, and heterolactic producing acids and alcohols. And that'll become important later on when we talk about different microorganisms that use these two different types of fermentation processes. This is the illustration that you looked at previously on another slide, but our focus this time will be on the right, where we're looking at the process of fermentation. Again, glycolysis takes place, breakdown of the 6-carbon sugar glucose into 2 3-carbon molecules, pyruvic acid. Then pyruvic acid, or a derivative thereof, basically goes through the process of fermentation, where you'll get the formation of